Yes.
Hello, everyone. Welcome to today's live broadcast, Quantitative Determination of Reaction Stoichiometry, Interaction Energies, and Solute Masses using Analytical Ultracentrifugation. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide window. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button. I would now like to introduce today's speaker, Dr. John Burgner. Dr. Burgner has over 50 years of experience with analytical ultracentrifugation, first using a Spinco Model E in 1963 in the laboratory of Frank Gerd at Indiana University Medical School. Dr. Burgner then earned a PhD at Purdue University for studies on the reaction mechanisms of lactate dehydrogenase, where he remained as faculty until retiring in 2009. In retirement, he volunteered his help to the biophysics department at the Virginia Commonwealth University Medical School, now serving as an adjunct faculty member. With such extensive hands-on training and experience on analytical ultracentrifugation, Dr. Burgner, over the years, has taught courses at the NIH and University College of London on Peter Shook's SedFit, and currently co-teaches Beckman Coulter's Proteome Lab customer training course offered here in Indianapolis. Before we get started, I would like to ask the audience a few poll questions. I'll let the poll populate here. The first question is, do you have an analytical ultracentrifuge in your lab today? Simple yes or no question. We'll let the answers populate, and we'll get right back to you. All right, we're pulling up the answers now. It looks like 60-40 split. 60% of you are saying no, and 40% are at yes. Our next question and final question will populate here shortly. For which applications do you use or would you use your analytical ultracentrifuge? Multiple answers are allowed here. Molecular weight, heterogeneity, molecular aggregation, interacting systems, molecular conformation, stoichiometry, or other. There's a wide variety of applications out there. Select the ones that you use the most. Just a few more seconds. Looks like the majority are using it for heterogeneity or molecular aggregation. You see that a lot in the biopharma industry. Molecular weight comes in at second, 22%. Molecular conformation, 16.2%. Stoichiometry and interacting systems tied at 14.3%. And 6.7% use it for other varying applications. So that's the extent of our poll questions. I thank everyone for your answers, and I'd now like to turn it over to Dr. Burgner for his presentation.
Good morning, everyone. This is John Burgner speaking, and I'm talking to you from Richmond, Virginia, where I currently live, although for many years, as he stated, I lived in West Lafayette, Indiana, and was at Purdue University. And so what I'm going to talk to you about today is a number of things, all of which relate to experiments that I have done over the past few years using the either an XLI or an XLA analytical ultracentrifuge. And these experiments have partially been for my own research and partially been for research of faculty members at Purdue and Virginia Commonwealth University, where I currently serve as an adjunct professor. So anyway, what we're going to talk about, as you see in the first slide, are applications which are commonly used for the analytical ultracentrifuge. I'm going to say AUC from now on because it's easier to say. Measuring solution mass or molecular mass of a particle, the heterogeneity, the number of different types of particles that you have in a solution, and conformation and shape, oligomeric. It should somehow got turned around, but that's not a particularly important use of the AUC in terms of measuring shape. Stoichiometry, where for instance have a heterogeneous complex, and we want to know what X and Y are for something. It could be A2, B2, for instance. The largest stoichiometry that I've actually ever measured was a system which you'll hear about today, where we had 240 proteins in the complex and 240 oligos of DNA. And then finally, association constants of the type 2A going to A2, or A plus B going to AB, and trying to determine an accurate estimate of what the dissociation constant is for A2 or AB. And one can go much more complex molecules as well. The other nice thing one can do, for instance, using Peter Shook's software, is that you can actually do ITC measurements. You can do light scattering measurements, ultracentrifuge experiments, and combine them in a global fit to get the largest or best answer that you can get. The kinds of samples that I've worked with over the years, of course, are proteins, nucleic acids, polysaccharides, just a little bit. They're hard to do. Small molecules. The smallest molecule, believe it or not, was air, and that was based on something that David Ufanis did many years ago, and I thought, maybe I can do that too. So we actually measured an equilibrium sedimentation for air, and then viruses and organelles, and as well as nanoparticles. Go to the next slide here. So the question that I've often asked students or postdocs when they come into the lab is, what do you want to know? And how much protein do you have, or how much sample do you have? And both of those are important questions. I mean, the size and the amount, and the slide has laid out a series of questions, difficulty as to whether the experiment that they're proposing is easy or hard, and how much sample they would need. Size and the amount of a component often can be a single experiment. May take 6 hours to do or 4 hours to do. Doesn't take much sample. We're talking milligram or less. Stoichiometry of a single component, is it A2, B2? That gets a little more complex and may take a little bit more protein. Is the reaction reversible? Well, if you change the concentration of the solute, And the sedimentation coefficient that you're measuring from a velocity experiment, again, 4 to 6 hours, will change, and that's indicative of a reversible process. The stoichiometry and energetics of self-association, now we're starting to get more difficult, and we may have to do a number of different experiments to get some information about stoichiometry and energetics. And then finally, stoichiometry and energetics of heteroassociation. That's even more complex, takes more sample. Now we're talking 10, 20, 30 milligrams of protein, for instance, and you have to collect more data, which necessarily makes it somewhat more difficult. Okay, so and now one of the things that I thought it was important to point out to people is, you know, with the XLA and XLI, you can actually make measurements from about 230 nanometers up to somewhere about around 600 nanometers. And I've got a picture of the spectrum of BSA there, and the two black arrows indicate the regions where I typically will look at the wavelengths that I will normally use in an experiment, either the 280 band or the valley at 250. Now, if, for instance, as we'll see later on, if you happen to use a sample which has, for instance, green fluorescent protein attached to it, then you can operate it at 290 or 393 nanometers, and you don't have conflicts with if you happen to have a nucleic acid present or a protein. You're just looking at the molecule that you want to detect. And the same way is that if you happen to have a fluorescein molecule, then you can operate at around 490 and be well away from protein spectra and nucleic acid spectra. Okay, so we have a number of different tools that we can use just from the spectral information of the sample that you're using. So the first thing I'm going to talk about, and I'm going to go back and say, well, I only did an absorbance run here. Well, actually, I did interference and absorbance. And this just shows a sedimentation velocity experiment where we have, and let me start the whiteboard here, and we have a series of boundaries across here that go, for instance, from left to right, and those are as a function of time and radial position. So each boundary is at a given time, which I think, if I remember right, these were 10 minutes apart. And then as you move down there, the sample is moving towards the bottom of the cell. We can analyze that data using the Lamb equation and using the CS distribution function of SedFit and calculate what's present. And we will get to that in a moment, but that's just one type of experiment. This is clearly a velocity experiment. In the next slide, I show a sedimentation equilibrium experiment with a small 12 kilodalton protein. And what you see is, starting at the top, the first scan up here, is that that's at time 0, and then at the last scan here at the very top is at time infinity, so to speak. And so at that point, you'll notice that the scans are getting closer and closer together, and so one is approaching equilibrium. Now, the interesting thing about this is that one is not limited to just doing a sedimentation equilibrium experiment or sedimentation equilibrium analysis. One can actually use all of this data here and treat it as a velocity experiment and calculate the mass. And this protein was about 1,280 kilodaltons, and when we did it, and we did this three times, is that we got within 100 daltons of the sequence mass for the protein. So it can be an accurate way of doing things. It's a lousy way of measuring the sedimentation coefficient because just the nature of the curves don't give you much information about how fast it's sedimenting. So anyway, and this is all I'm going to talk about the mathematics behind the sedimentation. And the principal equation is the Lamb equation, which is the differential equation, which is a function of the diffusion coefficient, which is forget what I'm doing here, diffusion coefficient here, and also S as sedimentation coefficient here, plus omega squared, the angular acceleration, and radial position, R, and the concentration. And programs like SedFit and Ultrascan and DCD Plus will make use of this in calculating sedimentation coefficients. Once you know the sedimentation coefficient, S, all right, which in the Svedberg equation, and also the diffusion coefficient in the Svedberg equation, then with the knowledge of the buoyancy factor, 1 minus V bar rho, then we can calculate the molecular mass of the particle. For sedimentation equilibrium data, we fit the data as an exponential, and there will be an exponential term for each solute that's present in the sample. What we're going to calculate is the mass of the particle once we manage to put in all the so-called fudge factors like the omega squared and the gas constant and the temperature and so forth. Okay? So going back to the sedimentation of BSA, which I want to talk about a little bit because it shows, in a way, it shows the beauty of the instrument and how accurate it is, and maybe a little bit about a problem that one has to be aware of, although it's not a very big problem. So, as I said earlier, and I guess my guess of 10 minutes was correct, that we have a series of interference scans here that, so this would be this is not the first scan, but this is, let's say, 30 minutes into the run, and then this would be, the second one here would be 10 minutes later, and so forth. And now we're looking down in this direction, this axis. This is radial position here. So, and now we fit this using what's called the CS distribution, which is a distribution of molecules with a given S value. So what we have here is a large peak, which is at around 4.3 S, and then there's two little peaks, one here and one here. And it's clear, well, it's well known, and the sequence is known and so forth, that BSA has a molecular weight of, I think, 68,000, something like that. If we analyze this data and determine molecular weight, it comes out to be a 68,000. So this is the monomer. So what are these two? Well, this turns out to be a dimer and a trimer. And one of the ways you can almost quickly tell that this is dimer, or at least give yourself an indication that it's a dimer, is if you take 4.3 and multiply it by the factor 1.5, then it's going to be somewhere about 6. And what that is saying is that if you take a sphere of mass 1 and a sphere of mass 2, the S values are going to be related by 2 to the 2/3 power, or about 1.59. So, and then you can do the same thing going from here to here, doing that, except it would be the 2/3 power of 3, which is 1.7 or something like that. Okay, so this is something that we're going to look at a little bit more in just a second. This is the experiment conditions we had. We had 0.1 molar sodium chloride, 10 millimolar tris, pH 7.4, and BSA was about 1.2 mg per mil. This is a very handy test for your centrifuge. We use this quite frequently just to make sure that everything is working well, because we know exactly how the protein should behave. This now is showing a comparison of 3 cells. For this particular experiment, I made up a batch of sample and such that I could fill 3 different cells with exactly the same sample and under the same conditions, and then ran them all at the same time. And this peak right here, again, is a monomer, and you can see for all 3 samples that the peaks essentially overlay one another. If you look here at the dimer peak, that's not quite true. I guess I should put an arrow. This is, again, the dimer peak. Now, if we go down here, this is a blow up of the 4.3S peak, and you can see that they don't overlap quite perfectly. And if we go and look at the dimer peak, again, they don't overlap perfectly. And what I was able to show, and also for the trimer for that matter, what I was able to show was that this peak, by doing several other experiments, that this peak here that is different from the other 2 actually is a function of the cell. We never could quite figure out what the difference was between the centerpiece for that caused this problem, but the experiment tracked, or this peak tracked, a particular centerpiece, and the other 2 were always gave peaks that overlapped. So, and you just cannot see a difference between the 2 peaks. Okay, we did this. Excuse me. We did the same experiment using the absorbance optics, and we again get a peak here for the monomer, and then a dimer peak here, and then a trimer peak, and maybe it's detecting some other peaks out here. If we go look at the next thing, the next slide, I have a comparison here between the BSA monomer from the interference data here, and then the absorbance data here, and the S values for these things are the same. Okay, unfortunately, there are different scales. Oversight on my part. If we go up and look at the BSA aggregates, all right, this is the interference data, and again we see that one peak doesn't overlay the other two, and with the absorbance data, it does overlap much better, and the reason for that is that instead of taking 20 milliseconds to collect a scan for interference data, it takes about 50 seconds of scan time to collect a scan for that, and so the sample during this period of time is moving, and that tends to smear things out or broaden the peak as the time goes on. Okay, so the lesson there is that you can get high quality data, it's reproducible, and that occasionally you will end up with a cell that doesn't behave quite, or a centerpiece that doesn't behave quite like the rest. Okay. All right, so going on now to what I was originally going to talk about is a series of experiments that I have done here at VCU, and this is on the adeno-associated virus Rep68 protein. It's a 61 kilodalton helicase that forms stacked rings at high concentration. So what I have here is a series of experiments at different concentrations of the helicase. So going from the lowest concentration here, where we have, and this is mostly because of resolution, a mixture of monomer and dimer here, and then we have a peak out here, which appears to be something much larger. And then if we increase the concentration, we now have a better resolution because we have better signal to noise, and we have now see the result of the monomer and the dimer, and then down here is very likely a tetramer, but it may not be, and then this is the large peak, which actually turns out to be a 7-membered ring. Okay, and as we increase the concentration, we get more of the ring, and this peak here also grows. Spectrum gets more complex as we get to even higher concentration, and we now have a new peak out here, and then finally at the highest concentration that we did, we have this new peak out here, which actually we think, although we haven't really proved the point yet, is that this is very likely a tetramer of the peak here. So this is a ring structure, and how do we know it's a ring structure? Well, we've done electron microscopy on it. That was supposed to be right here, but didn't get there for some reason or other. And you can clearly see the seven molecules in the structure, or 7 subunits. Let's put it that way. Here, what I've done is because one of the things we wanted to do was to get some idea what the dissociation constant for each of these molecules are. What I did was integrate the area under this peak, which gives me the amount of protein that is a monomer, and then if I integrate the area of the second peak here, that gives me the area of or gives me the amount of the dimer, and from the ratio of those two, I can estimate, because these appear to be these are not in dynamic equilibrium, or let's put it this way, they're not in fast equilibrium, they're on slow equilibrium on a time scale of the centrifuge, so these peaks are pretty stable. and don't shift with time or going down the centrifuge. So we can estimate now that the dissociation constant for this for the reaction 2 goes to 1 is about 1.5 micromolar, and 2 goes to 4, 1.8, and then the final one, which is where we close the ring structure, we have that this is going to 6, but it's probably not 6, is about 10-fold larger. And if you think about it, what is happening here is for the last molecule here, this is where we're closing the ring, and instead of one interaction between a monomer, 2 monomers, you now have 2 interactions between the 2 molecules that go together to form the ring, which you wouldn't normally expect this interaction to be stronger. So that is this one right here. The arrow going the wrong way. Okay. Now the other thing is this is a helicase, and so it binds DNA, and we are looking here at one of the oligos that bind to Rep 68, and this is the origin species from the viral genome, and what we see is that as you increase the concentration of protein in the presence of the oligo, is that you get an increasing amount of this peak that's about 12S right here. And the oligo at some point disappears completely, like in this experiment right here, and we only have the complex, and finally we have the complex here as well. And this turns out to be also to have 7 molecules in it, and we're trying to now relate the concentrations to get some idea of what dissociation constants are for these things, but we haven't convinced ourselves yet that we have. All right, so another experiment, again, that's related to this in a way, is this was a pure RNA binding to the bacillus pure R protein, response protein, and we got interested in doing centrifuge work on this molecule because the structure was done at Purdue by Janet Smith, and it's a tetramer. Well, what's unusual about a tetramer? Well, the binding site clearly for the RNA is in the middle here, okay? And there's no way that the pure R RNA molecule can actually fit in there. So the question is what is going on here? Is this dissociating to form a dimer, or is it dissociating to form a monomer, or what? Well, but it clearly indicated it can't be a tetramer, and there are other forms of this protein which actually appear to be a dimer. So we did, and the first thing we did was actually a velocity measurement, and for 2 different forms of the enzyme. One is his-tagged, and one of them is not his-tagged, and we wanted to be able to use the his-tag because we can get the protein far more pure, and because since we're using RNA, if we have any ribonuclease around, our RNA begins to disappear with time. So anyway, so what we wanted to do in this experiment here is we wanted to see if we could show that as you decrease the concentration of the pure R, that we would see a change in the dissociation constant, or the calculated mass of the particle as you decrease the concentration of the protein. And so the highest was over here and the lowest is over here. What we found was that there's no significant difference between the dissociation constant here and the dissociation constant at the highest concentration. They're all right at 90 kilodaltons. And now, actually, for the lowest concentration, the amount of sample and the amount of signal is pretty low there. But the software comes back and indicates that that this is also 90,000. The issue there is you have to be very careful in your analysis, or it simply diverges to meaningless answers. So okay, so it doesn't appear to be, it appears to be a tetramer. And from the velocity data over here, again, it appears to be a tetramer. The dissociation constant, or not the dissociation constant, the mass of the particle in both cases is around 90 kilodaltons, which is very close to the sequence mass. All right, so what did we do? Well, what we did was we started out with an experiment where we titrated, we used the ultracentrifuge as a very fancy titrimeter. So what we did is we titrated the pure RNA with the protein. So in the first experiment here, we have just pure R, okay, at 0.3 micromolar. And then we started adding protein, and the protein concentration ratio was, I think, 1:1 here and 1:2 the next one, and so forth, and then finally the protein. So you can see the protein is with an S value of 5, and it shifts in the presence of this is pure protein right here. Okay, and its S value shifts in the presence of the pure R. So what we did is that we integrated the peaks and showed that the concentration of free RNA as a function of the ratio of free RNA to the protein right here, you get a typical titration curve-like line, and that if we use our imagination and were to draw the line across like this, then it's going to give a stoichiometry about 2. We also went back and looked at the the masses of these particles, and it appears to be the ratio of 2:1 protein subunits to RNA particle. Now, you might say, well, why did you go to such extremes to do this? Because if you think about it, you've got 2 different species in here. You've got protein, which you can measure, and you've got DNA, or RNA, which you can measure based on absorbance optics, Why can't I just calculate that? Well, the problem when you're working with RNA or DNA or RNA oligos with proteins is that if the oligos have any size to them, that their absorbance simply swamps out the protein absorbance, and so you can't you have a difficult time analyzing the absorbance part. The interference part's easy because it's just the total mass. So anyway, we concluded that this is on the basis of 2 or 3 observations, and that the affinity based on doing gel shift assays of the RNA to the protein is in the 10 to 15 nanomolar region, which is beyond what we can actually detect in the AUC. Okay, all right. So the next thing that I want to discuss is a set of experiments that we did with Simbas virus. And the virus is a large structure. It's about 700 nanometers across here. It consists of a core of RNA. This is single-stranded RNA. It has a capsid around it that's made out of 240 individual capsid proteins, which you probably, and then it has a lipid membrane, which is the blue region right here, and that it picks up from the it actually picks this up from the host as it's being excreted, and then it has a glycoprotein outer layer which consists of E1 and E2. So, and what we had 2 questions we wanted. Well, the initial question is we wanted to know something about this core particle, because we can assemble this particle in vitro very easily with a 48 base pair oligo and a mixture of the protein, and get a what appears in the electron microscope as a sphere. And it's a homogeneous particle, as we'll see in the ultracentrifuge. And then one of the things you sort of can see here at the bottom is that there is a bridge from the outer, oops, wrong way, John, from the outer protein to the capsid protein, and it's thought that this is important for, and this thought that comes from mutational experiments, that this bridge across here is important for assembly of the outer core of the virus. So what I did was we did a velocity study. All right, this is the interference optics at the top. You can see it's a nice boundary that moves down the cell. It's a little broad, which indicates that there's more than one species present. We also labeled the, there are lots of experiments like this. We also then have a set of experiments where we labeled the DNA with fluorescein, and then followed the sedimentation and absorbance optics of the fluorescein down the cell. Well, why did we do that? We wanted to find out how many molecules of DNA, how many molecules of oligomer are bound per molecule of capsid protein. The virologist wanted to know this piece of information, and this is how we came up with doing it. And so what we did is we determined the total mass from the loading concentration in fringes for the particle, and that is the red line here. All right, and then we also estimated the concentration of dioligo, which is the green line here, based on the absorbance of the fluorescein derivative. And then when you do the calculation, you find that the concentration of capsid protein to DNA is approximately 1. I think it came out 0.98. So, but that again is 240 copies of the capsid protein and 240 copies of the DNA. Now we used single-stranded DNA here because it's more stable than the RNA, which is the RNA virus, because this is a single-stranded RNA virus. The other thing that we wanted to do was to look at the thing that's the peptide or that is going through the membrane, okay, and making contact with the capsid protein, and that protein is controversial whether it exists. I mean, the basis for it is mainly electron microscopy. I will say that if you mutate that, you don't get virus. You don't get a complete assembled virus. So we did two experiments. One, we did we did two velocity experiments. One we did with the capsid protein in the absence of RNA, and that's the top experiment. And go away. Up here, and so what you have here is this is the RNA. No, sorry, that is the protein. This is the RNA, and then this is this is the green. You have green fluorescent protein in here, and that is the one here. And over here we have the capsid protein, and the green fluorescent protein has the E2 peptide, the transmembrane E2 peptide bound to it. We mix them together, we put it in the centrifuge, and we get a new peak here, which is a mixture then, can be shown based on the mass, the calculated mass, is a mixture then of the capsid protein plus the transmembrane E2 peptide labeled with GFP. So clearly, under the right conditions, you can get a complex between these two species. If you don't have the E2 or the E2 peptide onto the GFP, you don't get a complex. So that was the control that we did. The other thing that we did was we did a velocity experiment. All right, here with the core protein. That would be just the DNA or the single-stranded DNA plus the capsid protein, and you get a peak. And the two peaks, one which would be the fluorescent or the green fluorescent protein peak, and wait a minute. Just a second here. I'm saying that wrong. Yeah. Okay. With the green fluorescent protein with the peptide label to it, and there is some fluorescence here, but we're not convinced that it is real. And it may only be just one or two molecules of the peptide bound to it. So this experiment here is not too informative, as we have actually, we're not working with it anymore, but it was not as conclusive as we would like to have seen. But the one at the top is clearly shows that the protein or the peptide GFP complex will bind to the capsid protein. All right, so this is another experiment where we're actually measured a small protein. And we did a sedimentation velocity at, which is shown by the experiment over here, And this is the distribution, down here, the CS distribution. So again, it's the distribution of molecules with a different size or different S value. And now what I've done on this side, and this is that I have done this experiment at 3 different concentrations. And you can see that that you begin to develop a peak here at a higher S value, which would seem to be the dimer. And so what you can do is you can integrate these peaks. So you integrate the entire peak, okay, and you can put this into Peter Shook's software as an isotherm, and do a calculation, and what you're going to fit is you're going to fit an S value for the, well, typically what we fit is simply the KD, because we know what S and so forth is for this molecule, for the monomer, and then do a fit. And you can see here that the data for the different isotherms over here, the different isotherms here for at different concentrations can be fit nicely with a model where you have a monomer-dimer equilibrium that is slow, and by slow on the time scale of the centrifuge. So we're plotting SW isotherm over here versus the concentration of the protein. The fit looks very nice, and we get a KD of about 270 micromolar. Well, the importance of this was simply to show, well, let me state it this way. The structure of this molecule had been done at Virginia Commonwealth, and but it clearly looked like that the protein was a dimer. But the contact area between the 2 monomers was not all that great, and so that's always a problem that crystallographers come up with. Is this contact meaningful or not meaningful from a biological sense? So we got a dissociation constant for this of 270 micromolar, and the conclusion was is this probably is not meaningful, but we don't really know. It's sort of in the iffy stage or state as far as that. This protein actually is secreted by a bacteria that lives in your gum lines, and it acts to inhibit immune responses against the bacteria. I thought it was really clever of the bacteria. And then the final thing here is that, well, I'm not going to talk much about this. This is again with the experiment we talked about earlier with the equilibrium data. You can go ahead and fit the data, and as I said, it comes out indicating that it is a 90 kilodalton tetramer. So the final thing that I was going to talk about is a velocity experiment, and in the top experiment here, we have we have velocity data for a protein at some concentration, and what I did is fit this using SEDFAT and determine a CS distribution plot for it. So now we have clearly two species here, one which is a monomer and the other is a dimer. And so the question is can we estimate a dissociation constant for this molecule? And when you try to do this, and we're now switching, instead of fitting individual CS distributions, we're now fitting it in terms of a model system of A plus A goes to A2, in other words, monomer-dimer equilibrium. So the information that we have to put in here is the molecular weight of the monomer, the molecular weight of the, or the S value for the monomer, which we can get here, and we can later calculate, and then we're to calculate a KD for this molecule, which is here, and then we're going to calculate an S value for the dimer. And so if you fit the data this way, then you get numbers which, in this case, this experiment is not very good because the data seems to have some problems. I did some additional things with the data but could never get a really great fit for this data. The one conclusion which I could have made up here on the basis of this data is that the dissociation constant, which is 3.4, is again in the 100 plus nanometer, not nanometer, 100 plus micromolar region, and so it's not a very strong association. So that is pretty much what I intended to tell you today and show you that the kinds of experiments that one can do and the kind of optical measurements that one can make that make, in some cases, back of the envelope calculations possible using the AUC and various samples. So if people have questions, I will be happy to answer them in the remaining 3 minutes and 14 seconds.
Thank you for that, Dr. Burgner. That was a great presentation. Before we get started on the question and answer session, I'd like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. We've had several that have already came in. Our first question is whether or not there is a limit in the mass of the sedimentation coefficient that you can analyze by AUC.
Yes, there is. There's really two different limits. It depends if you're working with a velocity system. I'm sorry, not velocity. If you're working with absorbance, then the limit is somewhat smaller than it is with interference, because you can make the measurement faster with interference than you can with absorbance. You can collect sufficient data to get an answer. The answer is in terms of size, and I'm going to give this to you in terms of sedimentation coefficient, is about 25,000 S that one can measure. Now, when you have 25,000 S, you probably are not going to get more than 2 or 3 scans from the instrument, and you probably won't see a single scan with the absorbance. So it's more how fast the sample sediments as opposed to actually the size of the sediment. If for instance you have a polystyrene bead which has a density very close to water, then you can have much larger particles in there than if you happen to have a gold particle which has a molecular weight of whatever gold has, but is dependent upon the size, and it will sediment very rapidly. And so there's a limit to how big the gold particles can be. That answer the question, you think?
Yes, definitely. Thank you. Our next question, there was a few questions that came in of sedimentation equilibrium versus velocity. When would you use one type of experiment versus the next?
Okay. I'm going to give you a smart aleck answer, okay? I personally would just as soon never use sedimentation equilibrium. I like sedimentation velocity, and I will explain why. One is that I oftentimes can get an answer on a problem in 3 or 4 hours by sedimentation velocity, and it may take a week by sedimentation equilibrium. I mean, if you're looking at a large protein, for instance, the experiment that I showed you using the pure R protein, which was 90,000 Daltons, each one of those lines, or each one of those scans took 40 hours to come to equilibrium. So that experiment, I could have, maybe there's an advantage of doing this. That experiment, I could have set it up, picked my speeds, put those in the centrifuge, and gone home for a week and then come back later. So, and gone fishing.
That sounds like a good use of your time. Our next question is whether or not an oligomer, which forms a soluble aggregate and comes out in the void volume in gel filtration, cannot be determined by AUC.
I don't have a good feeling for how large of an oligomer that won't come out in the void volume. But my guess is that, and I will say this, that we have done a few experiments with exosomes, which are fairly large particles, and managed to get useful information. We've also done viruses, which and gotten useful information. So, and these are large particles, millions molecular weight. So, my guess is, and my approach to this is always going to be, it only costs me a day or less to actually measure this or to actually run an experiment to find out whether I can get useful information. And you can always, if you don't have an AUC, you can always find somebody that will do it for you.
All right, yeah, thank you. Thank you again, Dr. Burgner. Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. John Burgner, for making today's educational webcast possible, which will also be available for on-demand viewing through beckmancoulter.com. Thanks again. Goodbye.